In the morning, citizens. Welcome to the next News and Guidance Special Report. Coming at you from an observatory in an undisclosed location, let's look at today's topic. Today, we are undertaking a deep dive into an idea that is, well, genuinely transformative. It really challenges the fundamental textbook view of human biology, doesn't it?
It absolutely does. We're stepping beyond the familiar world of hormones, neurotransmitters, electrical synapses, all that stuff.
And asking you, our listeners, to consider something pretty radical. What if the main way your cells talk to each other, coordinate everything, organize themselves? What if it's not primarily through chemistry?
What if it's through light? That's the core idea. We're diving into biophotonic signaling today.
Right. It's this proposal that the human body is, in essence, unified. One continuous, highly structured electromagnetic whole.
Exactly. And our mission here is to synthesize the foundational scientific arguments for this model. We'll trace it from the very first hints of ultra-weak light emission right up to how this system might organize consciousness, even systemic function. It's a big picture.
We're tackling those deep questions that, let's be honest, the standard chemical signaling models have always kind of struggled with. Like, how does genetic information get coordinated instantly across trillions of cells?
Yeah.
All at once.
Or morphogenesis. How does a developing body know exactly what shape to take? How does that intricate structure emerge?
The answer, according to the sources we're looking at, is this concept of electromagnetic unity.
It really forces a mental reset. I mean, if this model holds up, life itself is fundamentally an electromagnetic phenomenon, a quantum process that guides, maybe even controls, the biochemistry we're more familiar with.
So chemistry becomes secondary. The effect, not the primary cause.
That's the implication. It suggests information transfer could be instantaneous or near instantaneous, not limited by how fast molecules can float around.
Okay, before we illuminate the deepest corners of this research, puns intended.
Ah, nice one.
We need to thank our partners. Thanks our sponsor this week, a cute little mini labradoodle named George that lives in the neighborhood the kids call him Barney.
Adorable. Okay, so where does this luminous idea even begin?
Let's start there. Section 1. The discovery and definition of ultra-weak photon emission, or UPE.
Right. And this idea of electromagnetic interaction between living cells, it's actually not brand new. You've got to go way back. Early 20th century, a Russian scientist, Alexander Gurevich.
Gurevich, yeah, real pioneer. Between, 1911 and 1924, he did these fascinating experiments, often with onion roots of all things.
Onion roots. And he noticed something really strange. If you put one actively growing root near another one, but separated them with a quartz window.
Quartz being important because it lets ultraviolet light pass through.
Exactly. When he did that, the second root nearby would suddenly show an accelerated rate of mitosis. Cell division just sped up.
But, and this is the kicker, if he swapped the quartz for regular glass, which blocks UV,
the effect completely vanished. Poof. Gone.
So he proposed this biological field theory. He figured cells must be influencing each other, not chemically, but through some kind of radiation. He called it mitogenetic radiation initially.
But the problem was the tools just weren't there yet. The scientific community couldn't verify these incredibly faint emissions he was proposing.
It was too far ahead of its time, really. He couldn't measure the signal definitively, so the theory kind of got shelved. Dismissed as this unproven biological field idea because the tech and the theoretical biophysics weren't ready.
Precisely. It took a major technological leap, which finally came around in the 1970s. And that's where the German biophysicist F.A. Popp enters the picture.
Fritz Albert Popp. His genius was using these brand new, super-sensitive photomultiplier tubes, instruments that could literally detect single photons.
Game changer. Popp and his team started meticulously measuring this ultra-weak photon emission UPE, and their work confirmed what Gurevich had only suspected decades earlier.
That living things do continuously emit this incredibly faint stream of light. It's real.
And Popp gave us the term we use now, the biophoton. From the Greek, bios for life and phos for light or power. Critically, his research pointed to DNA as the primary source.
Right inside the cell, at the very core.
Which anchors the idea that this isn't just some random surface effect. It's originating from the cell's information center.
Okay, so UPE. It's universal, found in most living systems, bacteria, plants, us. It spans a wide light spectrum, from UV around 200 nanometers up to near-infrared at 800 nanometer. But the defining thing is the intensity, isn't it?
Oh, absolutely. The intensity is why it was missed for so long and why it's easy to just write off as noise. We are talking ultra weak.
How weak?
Sometimes just a few photons per cell per day,
Okay.
or maybe a few hundred photons across a whole small organism in a day. It's incredibly dim.
And this is where it gets kind of personal, right? For you listening.
Yeah.
You can't see this. We can't see it.
Nope. Our eyes are amazing detectors, incredibly sensitive, but there's still a threshold. You need something like 1,000,000 photons hitting a square centimeter every second. $106 text photons great for our brains to register it as light.
But UPE, especially in that visible and UV range below 700 millimeter, it's measured way, way lower. Somewhere between like 0.1 and maybe 10,000 photons per second per square centimeter, 10^1 to 104 o'clock.
Millions of times weaker than what we can perceive. So there's this constant, intricate light show happening inside us, inside every living thing around us, and we're completely blind to it.
And for years, if scientists did pick up this faint light, the standard view was just cellular exhaust, right? Random noise.
Yeah, maybe a byproduct of metabolism, oxidative stress, just thermal junk.
Yeah.
Chaotic, meaningless.
But the paradigm shift, the big change in thinking, comes from Pop and later researchers showing that UPE isn't chaotic at all. It has coherence.
Coherence, that's the absolute key. It means the light waves are synchronized, they're traveling in phase, organized, like a very, very weak laser beam, not like the scattered light from a regular bulb.
And that property, coherence, combined with other characteristics linking it to these specific wave structures called solitons, which we'll get into,
Right. It elevates UPE from being just cellular waste.
to being an active, organized mechanism for electromagnetic communication between cells.
It's a whole new scientific paradigm for biology. That coherence implies structure, and structure can carry information.
Okay, so if these biophotons are coherent carriers, that puts them in a fundamental role. Let's move into section 2, biophotons as fundamental carriers of both energy and information.
So the thinking goes, if they're coherent, they're not just waste. Biophotonics, the field studying this, proposes that photons are involved right down at the quantum level in organizing atoms. They structure matter itself and ensure the flow of all electromagnetic processes.
Which leads to that fundamental assertion you mentioned earlier, life is primarily electromagnetic. The chemistry we see and study is the result, the downstream effect of this underlying electromagnetic organization and control.
Yes. And this is where our picture of the human body itself starts to change quite dramatically. It brings us to this concept of the liquid light body.
Liquid light body. Sounds a bit out there, doesn't it? But it's actually grounded in condensed matter physics, right?
It is, surprisingly so. The body isn't just a bag of water. It's understood here as a highly specialized system of liquid crystal structures. And these structures have distinct semiconductor properties.
Okay, break that down. What structures?
Well, first, water. We're mostly water, 45–90%. But a significant chunk of that water isn't just loose H2O. It's thought to exist in a highly structured state, forming energy-intensive liquid crystals.
The sources mentioned a specific spiral structure, $300 and $100 and $201.
That's one of the complex structures proposed. The point is, this ordered, structured water isn't just a passive solvent. It shows weird biological anomalies. It can store and transmit energy in ways simple water can't. This structure is crucial for coherent signals to propagate without just fizzling out.
Okay, water. What else?
Cell membranes, lipids. They naturally exist in a liquid crystal state, too. Functionally, they act like tiny capacitors, building up and holding electric charge across boundaries. That potential difference is fundamental.
And then there's connective tissue, often overlooked, but huge in mass.
Huge. About 85% of our total body mass is connective tissue, think collagen, fascia. These biopolymers aren't just structural scaffolding. They have clear semiconductor properties.
Meaning they can conduct electrical signals under certain conditions.
Yes, and more than that. When you stretch or compress these tissues just by moving, they generate piezoelectricity, an electric current from mechanical stress.
So you put it all together. Structured water, liquid crystal membranes, semiconductor connective tissue. You get this highly ordered, crystalline-like medium throughout the entire body.
Exactly. And what happens when light photons travels through a highly ordered crystalline structure? It behaves differently than light in empty space.
How so?
The photons interact strongly with the material. They can excite electrons, forming these quasi-particles called excitons. And when these photons and excitons couple strongly, they form exciton polaritons.
Polaritons, and these are the liquid light.
That's the concept. These polaritons are quantum entities that share properties of both light, like moving fast, and matter, like interacting with each other. They can flow, almost like a liquid, through these biological crystal structures, hence liquid light.
Okay, if the body acts like this organized liquid crystal semiconductor matrix, allowing liquid light to flow.
Oh.
What's the really radical implication for energy transmission?
The radical conclusion, and it is radical, drawn from the physics of these systems, is that human tissues could potentially exhibit properties akin to superconductivity.
Superconductivity at body temperature. That sounds impossible based on conventional physics.
It does, but the argument is that these specific biological liquid crystal structures might enable it. It would mean instant transmission of electromagnetic energy, this liquid light, with virtually no loss to resistance or heat.
Which would explain the incredible speed and efficiency needed for biological coordination, right?
It's one way to explain it, yes. It bypasses the speed limits of chemical diffusion entirely.
Okay, that covers the efficient energy flow. Now, let's focus on the information side. Why are photons considered the ideal carriers for information inside a cell?
Well, simply put, they're the fastest things we know for carrying quantum information. Our entire global communication network, the internet, runs on photons traveling through optical fibers.
Right.
So inside the cell, the idea is that biophotons provide this real-time, instantaneous transfer of information about what's happening metabolically, in vivo as it's happening.
And the link to quantum computing highlights the sheer density of information they could carry. Qubits.
Exactly. Photons are prime candidates for making physical qubits. They have the basic polarization states, up, down, or left, right, which can act like a binary 0 or 1.
But there's more to it than just binary.
Much more. Photons also have orbital angular momentum. You can think of it as a twist in the light beam. And this twist isn't just on or off, it can have many different values.
So each photon can carry information on multiple channels simultaneously.
Potentially an infinite number of states, theoretically. This gives them an incredible information-carrying capacity, far beyond simple on-off chemical signals. It allows for a very dense information matrix within the cell's communication lines.
Okay, so we have the carrier, coherent biophotons, the medium, liquid crystal body, but we need a mechanism for efficient, lossless energy delivery over molecular distances. This takes us back to that crisis in biophysics you mentioned. Section 3. The Soliton model and DNA functioning.
Right. The crisis was about speed. The classic model relied on energy from ATP, the cell's fuel molecule, being passed along via something called pi electrons. But when you calculated the expected speed based on normal thermal dissipation and diffusion,
It was just too slow.
way too slow. It couldn't explain how fast biochemical reactions actually happen inside a living cell. The energy would seemingly spread out and weaken, dissipate as heat, before it could effectively do its job over longer molecular chains.
The cell seemed like it should be incredibly inefficient based on that old model.
Precisely. The breakthrough came from Soviet physicist Alexander Davidov and his concept of the Davidov-Soliton.
Soliton, explain that again. It's not like a normal wave that spreads out.
Correct. A soliton is a special kind of wave packet. It's structurally stable. It holds its shape and energy together as it travels, even through a noisy or nonlinear medium like a protein chain or DNA helix. It doesn't disperse. Think of it like a tiny, self-contained energy bundle moving along the molecule.
Okay, so how does the cell make these solitons?
The theory is that biopolymers, proteins, DNA act like converters. They take the relatively incoherent, messy thermal energy released from an ATP molecule breaking down.
And transform it into the coherent, organized energy of a soliton.
Exactly. The energy gets channeled into specific molecular restorations, creating this stable wave packet that can then travel down the molecular chain, say, along a protein alpha helix without losing its punch.
So the energy arrives at its destination intact, ready to power the next step in a reaction pathway. That solves the speed and efficiency problem.
It makes the soliton mechanism potentially the universal basis for energy and information transfer at the quantum level within the cell. And this brings us right back to DNA, which Pop identified as the main light source.
So DNA isn't just the blueprint storage. It's the central command center, the router for this whole biophotonic network.
That's the interpretation emerging from this model. DNA is described as an excited duplex, an exaplex system. Its double helix structure seems perfectly designed to receive and efficiently accumulate electromagnetic energy, basically trapping photons between its strands.
Now here comes the really mind-bending part. The sources suggest these trapped photons are held within the DNA in a state resembling a Bose-Einstein condensate.
Yeah, this is probably the most challenging and revolutionary claim in the whole model. A Bose-Einstein condensate, or BEC for those listening, is a state of matter usually seen only near absolute zero temperature.
Right, where atoms get so cold they lose their individual identity and start behaving like one single coherent quantum wave. They essentially merge. You need a cryotrap to achieve that.
So the obvious question is, how on earth could DNA create a BEC-like state at warm, messy body temperature, 37 degrees C? It seems to fly in the face of standard quantum physics.
What's the proposed explanation?
The theory is that the unique, highly regular geometry in the electromagnetic environment within the DNA double helix itself acts as a kind of structural cryotrap. It shields the trapped photons, or more likely the excitant polaritons, the liquid light, from the thermal chaos outside.
So the DNA structure itself stabilizes these coherent energy packets.
It captures them, compacts them, and essentially freezes them in time, locking them into a coherent, synchronized state protected from the cell's heat. This could also explain the incredible stability of the DNA molecule itself.
And if DNA is doing this, it provides a massive reservoir of coherent, perfectly aligned photons all humming at the same frequency and phase.
Which then throws a whole new light, literally, on the function of non-coding DNA, that 97–98% often dismissed as junk.
Maybe it's not junk at all. Maybe it's the quantum hard drive.
That's the speculation. If DNA is storing and stabilizing this quantum energy, then perhaps that vast majority of inactive DNA, together with this frozen biophoton energy, is playing a crucial organizational role.
Not coding for proteins, but orchestrating the when and where the coding parts, that 2%, get expressed, using this stored coherent energy.
Precisely. DNA acts as the master router. It receives environmental signals, stores coherent energy, and distributes the organized information signal, creating this constant, structured electromagnetic field inside and around the cell.
A kind of cellular Wi-Fi network, keeping everything in sync.
That's a good analogy. But like any Wi-Fi network needing cables, you need transmission lines within the cell and tissue. We talked about the liquid crystal medium, but what specific structures are thought to carry the signal?
Microtubules. Collagen.
Those are the prime candidates. The vast network of microtubules inside the cell and the collagen fibers forming the extracellular matrix outside. Their known semiconductor and crystalline properties make them ideal conduits.
Like biological fiber optics.
Essentially, yes. They can transmit, store, and distribute this coherent biophoton signal, originating from the DNA router, across the cell, and potentially across large distances within the body's tissues.
Okay, so let's scale up. From the cell to the whole organism. Section 4. Systemic organization and transport. How does this biophoton signaling integrate the entire body?
Well, the promise is that it offers a unifying mechanism. Instant coordination of metabolic reactions across trillions of cells. That's huge.
Makes sense if the signal travels at light speed.
Exactly. Then there's morphogenesis, guiding how tissues and organs form during development, how wounds heal, maintaining shape.
How would light guide physical structure?
Through the creation of coherent electromagnetic fields that act as organizing templates, perhaps, guiding cells to the right place, telling them when to divide or differentiate. And it might also solve the protein folding problem.
Ah, yes. How these long protein chains snap into complex 3D shapes almost instantly.
The idea is the existing coherent field guides the folding process, rather than it being just random thermal bumping until the right shape is found, which seems statistically impossible for large proteins.
And this happens through the interaction of individual cell fields.
Right. It's called field conjugation. The electromagnetic fields generated by individual cells don't just sit there. They interact, they overlap, they become coherent with each other in tissues and organs. This creates a larger, unified field state.
So the health of the tissue depends on the coherence of this collective field.
That's the idea. The sources even suggest that if a single cell dies, the slight change or blip in the overall resonant frequency of the tissue's field acts as a distress signal.
Triggering regeneration in the surrounding healthy cells.
Potentially, yes. An immediate field-based damage signal.
Okay, if cells are constantly generating and exchanging this coherent energy, we need a main power source for the whole system, right? Where's the body's central generator for this field?
The sources point clearly to one tissue type as the quantitative leader: striated muscles, skeletal muscles.
Muscles. We think of them for movement, maybe generating heat.
They make up a massive percentage of body weight, 40–50%. And critically, they contain the highest total number of mitochondria in the body.
More mitochondria means more ATP, more energy conversion.
Exactly. This model proposes a profound new role for muscles beyond just movement. They are seen as converting the chemical energy of ATP not just into mechanical force and heat, but significantly into coherent electromagnetic energy.
So muscles are the body's main quantum electromagnetic power plants, continuously generating the coherent field needed for everything else.
That's the hypothesis. Their constant low-level tone, even at rest, might be primarily for field generation, supplying the energy for this body-wide communication network.
Which immediately implies we need a way to circulate this energy, right? You said the liquid crystal body isn't great for just radiating the signal everywhere.
Correct. You can't just broadcast it efficiently. There must be physical pathways, dedicated conduits for the continuous circulation of this coherent electromagnetic current, this liquid light.
Biological power lines and fiber optics.
Pretty much. The first candidate system we've touched on is the myofascial network.
The interconnected web of muscles and fascia. We know fascia has those semiconductor properties.
And it's continuous throughout the body. Muscle chains, fascial planes, they form a ready-made network for potentially rapid transmission of these signals via solitons or other mechanisms.
But there's another, maybe even more compelling candidate structure mentioned.
Yes, and this one is fascinating. The Primo Vascular System, or PVS.
The PVS. This has a bit of a controversial history, doesn't it? First proposed back in the 60s by Kim Bong-han.
That's right, a North Korean scientist who claimed to have found this entirely separate circulatory system, thin, thread-like vessels and nodes distinct from blood vessels and lymphatics. He even linked it to traditional acupuncture meridians.
But his work wasn't easily replicated at the time, and it kind of faded out, got dismissed.
It did. The technology wasn't quite there. But fast forward to the 21st century. Work by scientists like Kwang-Sup So in South Korea, using modern techniques, specific stains, electron microscopy, injecting nanoparticles.
They actually found it, proved it exists.
They did. They've demonstrated the existence of the PVS in various animals and in humans, located exactly where Kim Bong-han described, inside blood vessels, along nerves, on the surface of organs.
Okay, so it's real. What makes its structure so interesting for biophoton signaling?
Its structure is remarkably similar to man-made optical fiber. It's got this distinct two-layer structure, an inner core, surrounded by an outer shell or membrane.
And the key is the refractive indices.
Exactly. The material of the inner core, let's say its refractive index is N1, is optically denser than the material of the outer shell, N2. So N1 is greater than N2.
That's the principle of total internal reflection. Light traveling in the denser core hits the boundary with the less dense shell at a shallow angle and gets reflected back inwards.
Forcing the light signal the biophotons to stay confined within the core and travel along the vessel with minimal loss. It's a biological optical fiber, perfectly designed to channel light.
Wow. And what's inside these PVS vessels?
A unique fluid called Primo liquid. And get this. It contains free nucleic acids, fragments of DNA and RNA, along with adrenaline, hormones, stem cells, immune cells like granulocytes.
Free DNA floating around in there, given that DNA is the proposed source and router of the biophoton signal.
It strongly implies the PVS is involved in transporting not just biochemicals, but also the electromagnetic information encoded or carried by that DNA and the associated biophoton field.
And this system appears very early in development.
That's another crucial point. Evidence suggests the PVS forms before the main blood vessels and nervous system during embryogenesis.
So it could be laying down the initial tracks.
That's the hypothesis. The PVS might establish the primary electromagnetic framework, the morphogenetic field that acts as the energetic blueprint for development. Subsequent tissues then organize themselves around this pre-existing coherent field network.
Mind blown. Okay, so we have the body's power grid and fiber optic network. Let's move to the central processor, section 5. Biophoton signaling in the brain and higher activity.
Right. If this system exists, the brain must be the most complex and intense generator and processor of biophotonic signals.
And there's evidence for that. The brain emits UPE.
Oh, yes. Studies have definitely shown the brain generates UPE. And importantly, the intensity isn't random. It correlates directly with the overall level of brain activity, with energy metabolism rates, with EEG patterns, even with levels of oxidative stress.
It's not just a correlation, though, right? There's direct experimental proof linking UPE to neural firing.
There is. For example, if you electrically stimulate nerves leading to the brain, or if you chemically induce depolarization in brain slices using high potassium to mimic intense firing, the UPE emission goes way up, dramatically enhanced.
And the opposite is also true if you block neural activity.
Yes. Using toxins like tetrodotoxin, which silences neurons, significantly reduces the UPE signal. This strongly suggests the light emission is intrinsically linked to the process of neuronal activation and information transfer, not just some unrelated side effect.
Neurons are these incredible integrators, right? A single neuron might get input from tens of thousands of synapses. Coordinating that requires something incredibly fast.
Absolutely. And the brain's structure might help. All those billions of neurons form this massive, complex system of electromagnetic generation. Their individual fields interact, and the skull itself.
Acts as a resonator.
That's the idea.
Hmm.
The skull cavity could act as a resonant chamber, helping to stabilize and perhaps amplify the collective, coherent electromagnetic field generated by all that neural activity.
And this resonance creates the brain waves we measure, alpha, beta, theta.
That's one interpretation. The brain rhythms are seen as macroscopic manifestations of the underlying coherent field dynamics. And what's really striking is they overlap.
Overlap with what?
The Schumann resonances. These are global electromagnetic resonance frequencies that exist in the cavity between the Earth's surface and the ionosphere. Some key brainwave frequencies fall right within the Schumann resonance bands.
Suggesting the brain might be naturally attuned to the planet's own electromagnetic field, using maybe similar soliton mechanisms for coherence.
It's a fascinating possibility, suggesting a deep connection between our internal biofield and the external environment.
Let's look inside the neurons again at the mitochondria. We said muscles have the most total, but neurons are packed too, and their mitochondria are specialized.
Very much so. Their shape and location change depending on where they are in the neuron. In the main cell body, they form this dense network like a power grid. In the long dendrites receiving signals, they often stretch out, elongated.
And in the axons, sending signals.
They tend to be more spherical, compact, and they actively migrate up and down the axon along microtubule tracks, going where the energy demand is highest.
And they pile up massively at the synapse, right, where communication happens.
They do. The classic explanation is simply providing lots of ATP for neurotransmitter release and reuptake, which is true, no doubt.
But the biophoton model adds another layer.
It suggests this high concentration of mitochondria at the synapse isn't just about chemical energy. It's also needed to boost the coherent electromagnetic energy, the biophoton signal, right there in the zone of interneuronal interaction.
So mitochondria, with their own DNA, act as local signal amplifiers and maybe even generators for the biophoton network, ensuring high signal strength for synaptic transmission.
That's the idea. Enhancing the density and coherence of the signal exactly where information needs to be exchanged between neurons.
And how does this sophisticated brain information get communicated back out to the body systemically? You mentioned the PVS has a nervous subsystem.
Yes, the PVS isn't just around organs. It runs within the brain's cavities, in the meninges, the brain coverings, down the spinal canal, and importantly, it travels along peripheral nerves.
So it provides a dedicated physical pathway.
A pathway for transmitting these coherent electromagnetic signals, the biophoton signaling, directly from the brain out to the organs, and crucially, carrying signals back from the body to the brain instantly.
A high-speed two-way street.
Exactly. And this, for the first time, offers a plausible, physically grounded mechanism for understanding psychosomatic phenomena.
The mind-body connection. How thoughts and emotions can influence physical health.
Yes. If thoughts and mental states correspond to complex, coherent electromagnetic field patterns in the brain, the nervous PVS provides the direct conduit for these patterns to influence the function of cells and tissues throughout the body, and vice versa. It translates the quantum coherence of mind into cellular reality.
Which brings us, inevitably, to the biggest question of all. Consciousness. Higher nervous activity. Our sense of self, the ego. Can this model offer any insights there?
Well, the sources tread carefully here, obviously, but they do propose a direction. If biophoton emission is ultimately rooted in DNA gene expression, and if specific neuron types have unique metabolic and electromagnetic environments,
Then maybe those unique local environments trigger highly specific, personalized patterns of gene expression within those neurons.
that's the hypothesis, leading to a unique, non-replicable signature of biophoton emission from each individual's brain, a kind of light fingerprint.
And this incredibly complex, personalized, coherent field filling the brain's quantum electromagnetic space is the awareness, the mind, the ego.
It's proposed as the physical substrate for it. The integrated coherent field is the consciousness. DNA remains the ultimate source and router, while structures like microtubules handle the transport and consolidation, much like the PVS and myofascial system do for the rest of the body.
That's profound. Okay, let's wrap up with the implications. Section 6. Implications and final thoughts, especially regarding health and disease.
Right. If biophoton signaling is fundamental to organization, then its disruption, its pathology, could be a fundamental new mechanism underlying disease, particularly non-communicable diseases, NCDs.
Also, take mitochondrial dysfunction, which is linked to almost everything: diabetes, heart disease, neurodegeneration.
According to this model, when mitochondria falter, it's not just a chemical energy crisis. It's a critical drop in the power and the information component of the body's biophoton signaling.
The signal density goes down. The coherence weakens.
Exactly. The signal gets faint, noisy. This initiates a vicious cycle. Cells can no longer properly perceive or respond to the unifying electromagnetic field. Coordination breaks down. Metabolic processes desynchronize. Molecular synthesis gets chaotic.
Leading progressively to cell pathology and eventually systemic disease. The quality of the light signal reflects the health of the cell's generators.
Precisely. So in summary, this whole concept potentially deepens our understanding of energy exchange. It gives DNA a crucial electromagnetic and informational role beyond just coding, and it provides a scientifically plausible mechanism for instantaneous, body-wide cellular communication.
The big takeaway really is, the human body isn't just a collection of disconnected chemical reactions. It's a single, deeply integrated, functional whole, united and coordinated by these sophisticated, coherent biophoton signaling mechanisms.
We are, in a very real sense, at the quantum level, organized fields of liquid light, coordinated by our DNA routers, powered significantly by our muscles, and networked by the PVS and fascia.
We've covered a lot. The liquid light body, Davidoff-Solitons stabilizing energy, DNA as a potential BEC router, the PVS as biological fiber optics, muscles as quantum generators, and that PVS link explaining the psychosomatic connection. It's a huge shift in perspective.
And if this is how we function internally, how might we leverage this for the future? How do we study it better? The sources suggest focusing on refining our analysis of existing tools.
Like MEG and EEG, magnetoencephalography and electroencephalography.
Exactly. Instead of just looking at the electrical signals or magnetic fields as indicators of where activity is, the goal would be to analyze the data to map the actual circulation patterns and the coherence properties of the electromagnetic fields themselves within the brain.
Moving neuroscience beyond just mapping activity into mapping the quantum field dynamics, that could truly revolutionize diagnostics and our understanding of the brain.
It's a powerful direction for future research. A pretty profound thought for you, the listener, to consider.
Indeed. We'll leave you with a piece of ancient wisdom that feels particularly relevant when exploring such deep and potentially transformative knowledge. From the book of Proverbs, chapter 10, verse 14.
The wise store up knowledge, but the mouth of a fool brings ruin near.
Adios, biophotonic emitters.